Salmonella can be classified into tymphoidal and non-tymphoidal. Tymphoidal causes tymphoid and paratymphoid fever. For the purpose of this presentation, we'll be focusing on non-tymphoidal, which causes gastroenteritis and food poisoning, specifically Salmonella enterica.
Salmonella enterica serovar typhimurium and its One Health significance. In humans, clinical signs include fever, diarrhea, abdominal pain, and sometimes vomiting. A paper published in 2011 stated the following figures for USA each year: 1.208 million cases, 19,000 hospitalizations, and approximately 400 deaths. In animals, in a study from 1996 until 2006, this subspecies had the highest incidence out of all Salmonella subspecies from isolations at Onestpoort and the agricultural research stations in South Africa.
Environmental contamination is an important factor to consider when looking at zoonotic potential in abattoirs. Salmonella can be carried into the abattoir in subclinically infected animals through their feces. Water is used frequently in the production line, meaning that infected water sources can also contaminate carcasses at any point in the process. Rumen contents can carry the infection, so contamination can occur if there is unhygienic handling of intestinal content on the line. This can lead to cross-contamination between carcasses on the line. Good hygienic and strict sanitation practices are very important in preventing the environment as a source of infection in the abattoir. During a study in Ireland in 2001 and one in Ethiopia in 2011, it was found that the equipment and work surfaces contribute to cross-contamination as frequently as the human or animal sources.
Plants are now regarded as alternative hosts for Salmonella. Eating fresh fruits and vegetables is an important way that humans can become infected. Water sources is mostly a problem in rural areas where cattle and humans share a water source. The cattle are able to defecate and contaminate the water that people drink. Raw or uncooked eggs is a major source of Salmonella infections in humans. Dairy products have also been implicated, mostly where the milk is unpasteurized or the parlor has a poor hygiene system. Lastly, animals may act as subclinical carriers, transmitting the bacteria to humans through direct contact, which could be from playing with your pet or feeding your cattle. The potential zoonotic ability is linked to the specific serovar.
Some negative health-related interactions at the internal microorganism molecular level is when Salmonella invades enterocytes and triggers intestinal inflammation, and this can subsequently then cause death of these cells, so intestinal absorption function is impaired. Then at the level of external species or organism, diarrhea leads to dehydration. The organism is undernourished due to malabsorption, and the normal daily functioning of this organism is then compromised. When it comes to systems, for example, an infection with Salmonella from contaminated water in rural areas where humans and animals share a water source shows an overlap in the ecosystems of humans, animals, and bacteria.
The main drivers of influence that result in each of the health-related interactions at each level are as follows. At the molecular or microorganism level, the severity of these infections depend on the serotype involved in the infection. In the case of Salmonella, almost all of the strains are pathogenic because they have the ability to invade, replicate, and survive in the enterocytes of the host, potentially causing fatality. The ability of the strain to survive in the host cells also play a major role, as those who are unable to survive are non-virulent. The causative of interaction at the species or organism level depends on the health status of the infected host. Generally, children below the age of 5, elderly people, and people with suppressed immune systems are much more susceptible to infection than healthy individuals. It is also more commonly seen in instances where these groups of people are housed together, as in the case of old-age homes or daycare facilities. At the systems level, many incidences are reported in people that visited African and Asian countries, thus importing the disease to their home country. This would be an example of systems that
Preventative
overlap.
actions on the molecular level target the microorganism. Heat and disinfectant can be used to kill the bacteria. However, freezing does not kill the organism, only decreases its numbers. On the species level, providing production animals and pets with salmonella-free feed is a preventative measure. Good hygiene management practices are of utmost importance, especially in high-risk individuals such as farmers, veterinarians, and abattoir workers. In retail, food control systems are put in place to prevent cross-contamination. With processing, practices such as meat irradiation combined with modified atmosphere packaging has been employed to prevent salmonella contamination.
The stakeholders are in the human medical profession, animal medical profession, water regulation authorities, agricultural companies selling food and feed products, and their provincial or territorial public health authorities. The World Health Organization is also in partnership with other stakeholders mentioned in the brackets in strongly advocating the importance of food safety by setting food safety standards. Now, all of these play a role in identifying and reporting the cases. In Canada, surveillance systems for foodborne illnesses, they use the following systems.
We found a study that was done to collect data from 13 European countries in regards to the incidence of plasmid-mediated quinolone resistance in Salmonella enterica. The MIC values of Salmonella samples collected from animals, humans, food and the environment was gathered from national reference labs and then screened for the quinolone resistance phenotype. PCR and sequence analysis were also done to ID the responsible gene. In the end, the gene was identified in 59% of the 1,215 Salmonella isolates. We can see the study involved the collaboration of multiple departments. To name only a few, a veterinary institute and a Department of Infectious Diseases in the Netherlands, a Scientific Institute of Public Health in Belgium, as well as an Agency of Environmental and Occupational Health in France. This is a prime example of people from different countries and professions working together towards